work. This is a really exciting time in the RNA field, and I encourage you all to look for as many opportunities as possible to get involved in RNA research, whether that is tackling complex diseases like cancer, neurodegenerative disorders, whether that's using our natural products that a lot of us work on to improve outcomes for patients with chronic non-healing wounds or for infectious diseases. This is a really exciting time to exploit the advances in RNA technologies. We're all aware of the mRNA vaccines and the impact that had in relation to COVID. There's a lot of work being done in that space for a number of other diseases, and hopefully we can capitalize on some of that by looking to develop collaborations with researchers working in that space around the world. Recently, there have been approvals of several small interfering RNAs, siRNAs, often for diseases where silencing of an enzyme in the liver can lead to improved outcomes for patients. And the seventh siRNA was approved just March this year by the regulatory authorities in America. This is also another area where we can look for ways we can collaborate to exploit siRNA technologies to improve outcomes for patients with complex needs in our communities. You'll also be familiar with the Nobel Prize for Medicine or Physiology being awarded to Victor Ambros and Gary Ruvkund last year for their work on the identification of microRNAs as key regulators of the human genome output. Well, they did the original work in C. elegans, the roundworm, but we've since come to discover that microRNAs fine-tune gene expression in many different contexts. This is an exciting time to consider how we might explore microRNAs as well, whether that's a case of seeing how natural products can modulate their function, or determining how we can use microRNAs directly transfected into cells to improve the outcome for patients. Another option would be to inhibit microRNA expression and function using small molecules or other compounds that we can inhibit the expression or function directly using antisense oligonucleotides, or we can think about inhibiting their ability to bind to some of their targets. One area that's also growing in interest is the use of RNAs as diagnostic and prognostic biomarkers for disease progression. And this is perhaps one of the areas where microRNA certainly first made commercial inroads through their utility and application to distinguish pancreatic cancer from pancreatitis, inflammation of the pancreas. If this is an area where you think there's opportunity to explore further, then do get in touch with me, because I'm working with a company at the moment to develop novel microRNA-based diagnostics for diseases. We're considering cancer, brain cancer in particular, at the moment, but there will be other cancers and other diseases to explore. So I'm only going to say a few words today, but I really want to encourage us to continue to build the RNA community across Africa, the RNA research community, and also to remember to take opportunities that come our way to engage with ordinary members of the public, whether that's in schools, in our local churches or mosques or other faith communities, in any community groups where we can have impact and influence, to really share the excitement about how RNAs are going to transform outcomes for patients over the next several years. If we have any influence in the policy space as well, then let's us, of course, try and reach out to our ministers and our politicians to ensure that they can buy into this exciting area. There is a challenge with a lot of the RNA-based medicines that have come to market recently, and that is the huge cost associated with them. Some of those costs can be justified in terms of the return on investments that the companies need, but actually the cost of making some of the medicines are really not that high, and we need our politicians to leverage their influence to ensure that we can receive such medicines in an affordable fashion for our communities, as well as develop our own homegrown technologies to deliver affordable RNA medicines for our countries and our communities. So without further ado, I hope you enjoyed the rest of the exciting session that has been lined up for you today. And I hope you take advantage of the chat box to ask any questions that you might have. And let's continue to build our RNA society communities for the greater good. And thank you for your attention.
That was a wonderful opening remark by Professor Kehinde. Thank you so much for your brief opening remark. I don't know if Professor Hyde is already with us, but please, Professor Hyde, if you are with us, can you indicate? If we wait a moment, then we can call on Dr. Prasad Filippo. Okay, I guess Professor Hyde will join us later. I've emailed him, maybe he's still busy with something. So before then, we have two guest presenter. Let me introduce to you Dr. Filippo Prasad. He has a background in biochemistry and molecular biology from University of Geneva, Switzerland, with a final internship in Oxford, UK. After a break into private field, clinical trial management in Geneva, then scientific consultant in Budapest, Hungary. He obtained PhD in plant biology. Following graduation, he worked at Thermo Fisher Scientific for 15 years as a sales representative, and finally joined Lexogen. Of course, Lexogen is one of our sponsor, our support company. Then finally, his role currently at Lexogen is a product manager, where he helps to bring new solutions to market, and latest addition of his efforts being Luto product family, which is going to introduce to us. Dr. Prasad, we are honored to have you. Please have the floor and let us get your presentation. Thank you.
Thank you. It's really also an honor for me and for our company to be invited to the RNA Salon. You're absolutely right. We sponsor this RNA Salon since many years. Before I joined, I only joined Lexogen 3 years ago. And we really try also, and Dr. Guinderos, also you mentioned it, to promote also all the science around RNA. So of course we are a company, we develop kit solutions, so it's for profit, but we also like to diffuse as much as we can some knowledge and to share it also among researchers. So today my presentation will be quite short, also due to time, of course. I will try to stick to the time. It will be an overview of the main challenges of RNA research, and also I will talk a bit more about our portfolio. If then you have questions during this conference or later on, you can also reach Lexogen later on, and we can also deep dive into more complex maybe products and applications. So I hope that my presentation will be useful. I will start sharing the screen in a few seconds. I just need to find the right screen. I'm on a few of them here, and this is it. Now I'm just sending a request for sharing, so I think you need to accept. Yes, and now it works.
Sure, I have accepted.
Excellent, thank you very much. Yes, perfect. So that's it. You should see my screen now.
Yes, clear.
Excellent, and we can start. So yes, I will just introduce Lexogen, some general, let's say, guidelines we have defined, and then the portfolio. Of course, the presentation will last about 15-20 minutes maximum. If time allows, some questions, otherwise maybe at the end of the conference or in further chats. So what is Lexogen? Lexogen is a company. We see here a few pictures that, and actually the people you see in the pictures are the real employees. Can you hear me? Is it, yes? Good.
Yes, yes.
Yes, thank you for confirming. Yes, yes. So we have been founded a long time ago, already 18 years ago, and this was in Vienna, in Austria. We have then received also some certification, especially the ISO 13485. This is an ISO certification that grants the possibility to our company to produce solutions, so kits, reagents, that will then be used for diagnostics. So we cannot make diagnostic kits, but we can make the materials that someone else can use.
Yes, it does be.
Sorry, can we mute our microphones, please?
Working on it from this end. Don't worry, I'll regulate. Thank you.
Thank you.
Thank you very much. So we are acting as a global company, either through distributors or direct. For African countries, it would be direct, but then you see, depending on the country, we can have different partnerships also with distributors. We have a subsidiary also in the US. That's why you have this red sign in the US here on the East Coast. And really, as I said, also, it's really important for us. Our mission is really to help researchers finding the right solutions for their needs. And for this, we really, let's say, we pride ourselves by providing a very dedicated, very specialized technical support that often receives 5 stars. But most importantly is really this technical support is here before even you purchase a kit from us or a solution or whatever. So you can really you have your problem, you can contact us and we can try with the help of tech support to help you, and of course afterwards as well. So there are 3 different proposals to work with Lexogen. Is either you can purchase products from us. So these are kits that can also be customized, by the way. We also can provide services. In this case, you send your sample to us, which will be in Vienna, and we basically perform the experiment for you. It can be a whole project, but it can also be a pilot project, and then you continue the project in your lab once you are a bit more maybe reassured that the technology was the right one, the strategy was the right one or not. So it decreases a bit the risk. And partnering, this is also something we do. So we like to partner, so to acquire new technology, to outsource new technology, so we can also create collaborations, many different kinds of them, by complementing some technologies together. And the workflow, in a nutshell, is this slide. So this slide shows really what we do. So we say we are RNA experts, but what does it mean? It's mainly dedicated to a next generation sequencing. So our core expertise relies in the NGS library preparation with all these kits that you see, and also in preparation of the RNA before the library, and also afterwards, which means bioinformatics, and we have additional products that I will mention a bit later. For services, this is a part of our service team. These are real colleagues in Vienna, and we provide basically from the start, from the consultation to the end, data analysis. We also provide services for data analysis, the whole workflow, and you can choose at which stage you want to pick. Usually our clients, they ask the full solution, but you can also stop. You say, okay, I have extracted my sample, you prepare the library prep, you send me the data, and I analyze the data. So you can really pick any of these steps with us. And here, so I will share, by the way, this presentation after the conference, so you can take the time, because it can be quite full of information and maybe you want to know more. In this case, for services, I encourage you to go to our dedicated webpage that then lists the different service types we offer, and then you can click on each tile to see exactly what it entails. Now, if you want just to go over a few concepts that are important then to make you understand how we tackle these concepts and how we develop the technologies. So this is coming from a review where the review DOI is here. So where a very important question before even you start the experiment is what we call the pre-analysis. So the design of the experiment, of course, the design of the sequencing strategy, of the controls. All these are questions that are crucial for you, but also for us to develop the right tools. And in this respect, these are typically questions that need to be answered before even you start the experiment. So basically, what is my research question and what type of data? Very important, because sequencing can generate a lot of diverse types of data. What does my sample type impose in terms of restrictions? And do I have any way of controlling what I'm doing? I throw a sample into a kit and then into a sequencer. How can I be sure that nothing went wrong, basically? So these are many questions that we ask ourselves. And part of the answer, you can read them also in our lexicon. The lexicon is a kind of manual that gives guidelines on many, many different aspects. So here you see a QR code that you can scan if you have a smartphone now, but you can also scan it later, and it gives you access to one of the chapters of the lexicon that specifically tackles this aspect of design and planning of the experiment. So I mentioned one of the questions, what is my research question? And what basic questions need to be asked like this, like, do I want qualitative data, want quantitative data? So do I want to know everything about my genes, or just to count genes, for instance? And this is where we come with a first split that then will determine which Lexogen solution you can pick. It can be Lexogen, it can be another one, of course. So whole transcriptome sequencing will be more qualitative, so it will go through the whole length of a gene, but it will generate more complexity. One gene, you will have a lot of different fragments, which means also more sequencing depth, more bioinformatics analysis. Sometimes it's needed, sometimes not. And when it's not needed, you can simply identify a gene with one fragment. And in this case, the method that we actually sell since a long time, one of the first ones, is really this 3' mRNA sequencing, which basically only generates one fragment of a gene, but in a sufficient length so that you know which gene it was. And the main application is to count the gene. And these are the two main solutions, the families of solutions. Then I will describe you the new one that was mentioned at the beginning, the Luthor, where Coral is a family of kits that covers all the genes. So cover all, that's basically the origin of the name, from the 5' UTR to the coding sequence to the 3' UTR, while the QuantSeq technology will cover essentially, this is the coverage percentage, so you see really a peak here in terms of coverage. QuantSeq will really focus on the 3' region. QuantSeq will be enough when you just want to identify and count genes. Coral will bring you much more information, but will add complexity and cost. So these are typical experiments you can do with abundance, treatment, control, which one is overexpressed in a cell compared to the other one. This is typically QuantSeq, expression profiling sequencing, and for Coral, a gene, as you see here, can have a lot of exons, of introns, of splicing variants that are very different in different cells. And Coral will really cover the full length of the gene, including the UTR, up to really both ends. It will also be able to cover something that is not a gene, so typically long non-coding RNAs. Something else, another question that I mentioned at the beginning is, what sample type will I be working with? And some sample types require specific, let's say, adaptation of the protocol, also of the sample preparation. And here as well, I don't go into detail, but I give you the web pages that you can read. Of course, we can also answer any further question, but we really focused on blood that requires specific adaptations. So basically removal of globin, FFPE. Dr. Ross, you mentioned cancer. Of course, FFPE is also something big and growing, where we have all these FFPE slides from patients that can be stored for decades. and they still contain RNA, and you can still extract RNA from them and obtain valuable information. But of course, it requires a special treatment, so it's not that obvious. But also for here, we have some specific solutions, and for plants as well, we also developed specific tools that combined then with Coral, with Quantic, allow you to handle these samples in the best possible way. Something else that is very general to any procedure is the control. The control needs to answer the basic question of, is anything going wrong at any step, including data analysis? And if yes, how can I detect it? And to answer the question, we have developed the SERV. These are spike-in RNA variant controls that, as the name implies, these are RNA molecules artificially produced by us and also designed by us that you basically spike into your sample as a control. And then to detect them, you will upload basically a data set that we call the servome. That is, it's an artificial data set, so we don't have any interaction with any other native RNA type, otherwise you would not detect them, where you can compare this serve depending on the sets that you choose for isoform bias, for dynamic range evaluation, or also for long reads, independently of the organism. And so the idea is this, is that you spike in, so you add your sample, your RNA control, the serve, into your sample, you process the sample until the end, and then you compare against the servome, and you can really evaluate if anything went wrong. It can be biases in the coverage, so a region is sequenced more preferentially than another one, or in terms of quantity, maybe you will lose part of the RNA. This you will also see because you know the amount of serve that you have spiked into your sample. So this is something very interesting that you can also use for any approach. Now, if we go to our portfolio, I will not describe it all, but the main solutions, so here you see the names, just maybe for sampling something that can be interesting, that was also described in the literature a few years ago. It's SLIME-seq. So this is metabolic RNA labeling. It means that basically you will label the newly synthesized RNA in vivo in your cells. And then you stop the labeling, you sequence, and you check which sequence was labeled and which one was not. Sequences that were not labeled were already present before the labeling happened, and sequences that are labeled are the new ones. You can also do the reverse way. So basically you label and then you sequence at a later stage where you can compare the disappearance of the labeled RNA. And this is what we call kinetic RNA sequencing. So it works essentially like the drawing shows, labeling of cells, and then the nascent RNA is labeled, then you apply some chemical treatments that end up in mutations. And by comparing the mutations against the original sequence, you have the possibility to quantify how many new molecules were synthesized or how many were degraded. So this is a method that goes at the beginning. So it is typically combined with, subsequently, with a Quantic method. SLIME-seq you label, Quantic you do the library preparation. I skip this one, it's quite simple. The split, this is a relatively basic method, but that we improve so to obtain really very pure RNA. It's a column-based kit where you can purify long fragments, short fragments. You can purify them all together or split, that's why the name, into a short RNA fraction, less than 200 nucleotides, down to 17. So you really capture even very, very short sequences and long fragments. So that's what this slide basically explains. And this is for general use of, you can extract basically any sample type. I mentioned plants, some adaptation for blood, and there's a new one for FFPE because FFPE is really special. You have to deparaffinize, you have to decross-link. So now we have a new split kit that also simplifies the workflow compared to existing methods. So it makes the workflow quicker. and still maintains high purity. Trapper, this is something very special and really that many customers who work with small RNA like a lot. It's a kit that will isolate, in particular, microRNA and small interfering RNA, but only the functional ones. The way it works is described here. It's an ion exchange chromatography that lasts a few minutes, where basically the small RNA, like typically the microRNA, that are bound to the Argonaute protein, part of the RISC complex, they flow through the column, while the non-functional ones, so the Argonaute bound are functional, the non-functional ones are retained on the beads. And therefore, afterwards, you have to basically separate the Argonaute protein from the captured RNA. You only purify small RNA and functional RNA. Then a more general consideration, again, one of the main questions that you need to ask at the beginning. So this is not valid for QuantSeq because QuantSeq will purify polyadenylated mRNA by default. This more applies to Coral. Should I choose poly-A selection or ribosomal RNA depletion? So this is really an overview. So if at some point you want to go into more detail, we can. Poly-A selection, as you know, probably know, this is the RNA Salon, is total RNA. When we speak about total RNA, it can mean many things, but usually it means a big mixture of RNA types. And among these RNA types, ribosomal RNA make the most of it. And usually you are not interested in ribosomal RNA. So what is possible is that you specifically, positively select the polyadenylated RNA, which is in majority the messenger RNA, the gene coding RNA. And this is one of the ways of preparing your sample before you go to the NGS library prep. The other possibility is that rather than positively selecting, you apply a negative selection. So basically you deplete the ribosomal RNA, and then you keep all the other RNA types. So it's not only the mRNA, it's many RNA, among which the long non-coding, the transfer RNA, small nuclear, circular, and other types of exotic RNA. Then, of course, you need to have a kit or a solution, a workflow that you can also design yourself to prepare libraries for this specific RNA type. So it's not always that obvious, but at least you have all of them by ribodepletion. So this is really a choice. And whatever your choice, we provide solution for both. For ribodepletion, we provide a specific solution that we call RiboCop, and that can apply to many, many different species. So also ribosomal sequences, even to globin, which is not related to ribosome, but it's a similar problem. In blood, you have a lot of globin and you are not interested in it because you want to see all the other genes. And for a RiboCop, we keep expanding actually the family by adding more and more and more species. So if at some point you also have a species of interest, I don't promise that we will develop a solution for this species, but please ask us, because we may have solutions, and sometimes there are also cross interactions. For instance, fish and plants, of course, it applies to most fish types and most plant types. So this is also the new addition. For poly-A selection, this is a universal module. Poly-A is poly-A, and that's it. You don't need to be species specific. QuantSeq I already mentioned. Coral, so this is for long read. And this is the newer one. Peter, you said it's a kit I worked on. I mean, I'm not in the lab. So product management is to make the bridge basically between the lab and the commercialization of the product. Luthor is we can say it's an extension in terms of application of the QuantSeq. It's a 3' mRNA-seq kit for now. And so basically targets the 3' end of a gene. And it increases dramatically the sensitivity. So it's ideally suited for 100 cells down to the single cell, 10 picogram of RNA. And I write N below because we want much, much lower than that for specific applications that are less than a cell. We went to 1% of a cell, so to less than a picogram, no problem. We can really detect all the RNA molecules that are present. The method used is different from QuantSeq. but the output is very similar. Even the analysis is the same. It's the 3' of the and then how to choose between Luthor and Quantic. Yes, it's basically about the input.
I think so. From the timetable is like 2 now. The follow up one. No, yeah.
I don't know if there is a question.
Yes.
It's okay. Thank you. And feel free to ask questions also through the chat, which I don't see, by the way, but I will see later. So how to choose between Quantic and Luthor is essentially the input. So here I added the input range, and then at 1 nanogram, of course, you can choose both. But of course, Luthor HD will see more genes than Quantic, which also means that Quantic does not see all the genes. And that's normal. And that's normal for most of the kits you see and most of the protocols, where low copy number genes are very difficult to catch. But with Luthor, you catch even 1 copy of them. And then this is just to show you that we also offer then derivatives of each kit. That's why sometimes I say family. Luthor or Quantic or also other kits have different kits. For this specific 3' RNA sequencing, you can handle your sample as a single sample, or you can also, if all the samples are quite similar, because that's a bit the restriction, you can pool all the samples up to 96 into 1 tube, which simplifies a lot the workflow. And we have this for Luthor and for Quantic. Small RNA, this is a new kit, 21st of May, so a few weeks ago. It's a kit that we call Mirvel Discovery. That's our most sensitive microRNA library preparation kit that we just launched with UMI, with UDI, so to really count genes and to discover, as the name says, even rare microRNA type sequencing. While the small RNA-seq library prep kit, this is the name, has been also in our portfolio since a long time. So this is not a new kit, but it's more for routine use, maybe not as sensitive as the Mirvel, but it can also work very well, especially with plasma that typically does not contain much RNA, in this case, microRNA. And then, if we want to summarize, if your initial question, remember the initial question is, I have enough information if I've performed 3' RNA-seq, then the choice is simply Quantic or Luthor, depending on which sensitivity you want to achieve, depending, of course, on your input. If you want to cover the whole gene, then Coral allows to cover the full gene, and then you just need to choose between poly selection. In this case, you will really essentially focus on messenger RNA. You can capture a few other ones. And RiboCop, you can also study non-coding RNA. This is because Coral can also prepare libraries from this non-coding RNA and a few other types, by the way. For small RNA-seq, we have our workhorse small RNA-seq kit, and then the new Mirvel for really the highest performance. And that's it. I think I come to the end of the presentation. I hope I was not too bad with the time. So if there is time for question, I can answer some now. Otherwise, I also stay online after the conference.
That was a wonderful session from
Thank you,
Professor Filippo. Please, you can put some reactions for him. Thank you so much. We will go to Q&A while we wait for Professor Hyde. I don't know why he has not been able to connect. So, but I would want to, before we proceed, use this opportunity to recognize the presence of Professor Kehinde Rose, University of Japan. He was one of the persons that make some recommendations for presentations. Thank you, Prof, for joining us. We also have the DVC of University of Agric, Umuagwo in Imo State, Nigeria. So thank you for coming, Dr. Obinna. So I would want us to go straight to question and answer so that Dr. Filippo will guide us and give us all the insights we need. Before now, some persons have submitted questions. So those questions you submit, you can also ask the question now. And we want to make it a discussion. So as we ask question, we also discuss. So if you have question, you can always raise your hand up. Then I will call you so that we flow in the same pace. Thank you. So Professor Kehinde, you can proceed with your question. Thank you.
Okay, thank you. Someone has their mic on in the background, which is causing a
Yes,
bit of
I'm working on it.
fantastic.
I'm working
Super,
on it.
thank you. Filippo, that was a really lovely overview of the Lexogen services and technologies and products. So really thank you very much for that. I guess one question for me is thinking about blood and biofluids, which would be better if I was interested in developing RNA-based insights to help with diagnosis or prognosis in cardiovascular diseases in particular. So would it be the Mervel tool to focus on microRNAs, or would it be the whole blood RNA-seq? And I guess maybe there's a cost consideration there as well, but any insights would be helpful.
So for blood, do you mean you only want to see the small RNA, or would you like to also see coding genes? Or
I guess because I have noticed recently there has been emphasis on cell-free messenger RNA in blood.
yes.
So, but I'm not sure whether that is something that's where the investments perhaps can be justified compared to just looking for smaller RNAs.
Yes, I really appreciate your question, because it's also a question we are working on at Lexogen, and it's also an important. Well, scientifically speaking, it's important, and then of course for our company, commercially speaking, it has to, because we need to invest quite some resources if we believe that's a promising field. My personal feeling is that yes, it's something that is starting now, cell-free RNA. For now, when you hear about blood, is essentially cell-free DNA, and for different reasons. One of them is the stability. DNA is stable and is well, annotations are also not quite mature, especially in human, because these have clinical applications. So you extract blood, you have DNA, it's easy to store, and there are many, many solutions out there to analyze it. But more and more, I hear about other researchers in academia, in hospitals, even I start to see some companies that really are interested now in cell-free RNA. So then you have to split between, of course, the messenger RNA and then the small RNA, typically the microRNA. So there is no single solution for both. You cannot prepare a library for NGS for both together. It would be nice. For instance, we would have loved, and maybe at some point it will come, to make a Luthor kit for microRNA. This would have been fantastic, because sometimes you don't have just enough material to prepare the library. Now with the Mirvel, also with the smaller RNA kit, we can go to quite low amounts, but still there is always the question, am I seeing everything, or am I missing something important? And then it's the question about low copy. If I have really low copy numbers of microRNA, but also of mRNA, do they matter, biologically speaking? Are there good biomarkers, or is it junk? So far, we just consider also during analysis as junk, because it perturbates a bit the analysis. So when we do the analysis, usually low counts are excluded, because then they can add a lot of bias. But they may be important. And when we developed Luthor, we noticed that in some cell types, especially cancer cells, more than half of the genes we saw were present in less than 10 copies per cell. Why are these genes there? We don't know, but we found them. And until now, no one had seen them. So this is a big question. Yeah. So and then the other problem is blood itself and the stability of RNA. We mentioned it. So we are really working on some solutions now to try and really work with small amounts of blood and with stabilizers. So there's one product I didn't present. It's not used for blood for now. It's the Defender. It's our first stabilizer, but we may come up with other one. So yes, to your first question, we believe that something is happening around cell-free RNA in blood, and that will hopefully take off in the next years, because RNA really brings something new, complementary, that DNA cannot bring. And especially in cancer, because cancer evolves, the DNA will be in general quite the same, not always, because you also have mutations, but the RNA changes much more quickly. And it would be super interesting to see what's happening at the RNA level in the blood, in cell-free RNA or in CTC, so isolated cancer cells from blood. And this may be very interesting, because maybe the cancer is not visible any longer. Maybe it's coming back, but we want to see it as soon as possible to apply the right cure. So yes, we're working on this. We're super interested, and we hope to find a solution soon. Actually, I cannot disclose more, but yes, we are really actively
There's more
working
going on, clearly, for what you are saying there. That's really wonderful. I guess one quick thing then, with the whole blood RNA-seq, does it capture small RNAs at all, or is it only messenger RNA and long non-coding RNAs?
So if the classical protocol will not capture small RNA, yeah. And it also depends on the preparation. When you extract the RNA, you need a method that does not remove the small RNA. So typically a method like split or similar, where you split the small and the large ones, this is the ideal, because then you use one library prep kit for the small ones and one library prep kit for the large ones.
Okay. And when you, with blood, is it serum, plasma, or either of those, or whole blood typically?
You can work with any type. Yes,
Right,
it doesn't matter so much. Again, then preparation can matter. And yes, ideally, you wouldn't even need to extract RNA from blood. So methods, and this is maybe the future, methods where you just draw your blood, a small volume, even at home, this would be ideal. You don't need to go to a hospital all the time. Whole blood, you ship, and we do the prep immediately from the whole blood. This is something that would be super nice, and yeah, that we would like to develop at some point, yes.
That's helpful. Thank you very much. Thank you.
You're very welcome.
Your questions, and the answer was insightful. Please, I call on Dr. Deus Dedit Subira to answer to ask his question.
Thank you very much. I'm in Imbarara. Maybe I can switch on the video since I'm asking questions. Yeah, I'm here in Imbarara, Uganda. I'm happy for your presentation. I have one question. It's also to do with the sample. I wanted you to share your experience with the samples, including maybe other body fluids, like people are talking about saliva. Someone I know someone who was interested in urine, looking for cancer, specifically cervical. So can you share some experience as far as such samples are concerned?
Yes, we have, especially our services team has been working with many biofluids. So urine, yes. Saliva, yes. These samples basically do not pose a lot of problems as long as they have been well preserved, because even though we are capable of extracting, so in this case we really extract RNA, yeah? So there is this extraction step. Sometimes if the RNA is degraded, then that's it. We cannot work further. Now with the Luthor kits and some other protocols that we are developing, so degradation is always a problem, because if it's degraded, basically we cannot work reliably with the sample. But input now is less of a problem, because we can really go to very, very low input. And so, yes, definitely, these samples are very useful. Then it depends what's in the sample. So, but they can contain, of course, RNA, and we can work with them. And they are less invasive than blood. So they are easier to work with, yes.
Thank you. Lugamba Nasif, please, you can unmute yourself and ask your question. Thank you.
Okay, thank you very much. I'm glad to be here. My name is Iduvanga Nasif. Actually, I'm graduating this June from Nanjing Medical University, a master's in clinical laboratory diagnostics. I'm from Uganda, and I look towards doing a PhD in diagnostics. This is so interesting to learn about Luxogen. Professor Philip, you slightly talked about the question that I had. Based on the high need for point of care testing, like precise diagnostics, you talked about like extraction-free methods, and you said that maybe this is where we are going. And actually, that's where my point was. Like, I looked at the protocols, and the protocols that you shared, I realized they all need processing. So I was coming there, like, can we look? Do you have a protocol? Because perhaps I'll do my study, and I was looking at a related study that maybe I use methods like ultrasonication just to break the fragments, and I also use maybe I make use of exonucleolytic activity of certain enzymes. For example, I was looking at some method called the rolling circumplication, where they use 529 polymerase, just the exonucleolytic activity of 529 polymerase. So I was looking at such a study, and I felt like this is something that's really important, especially in point of care testing. I wonder whether you have something you can advise in that line. Thank you.
Yes, thank you for your question. So yes, when it comes to diagnostics, so we don't develop diagnostic solutions ourselves. So it's probably this kind of solution may exist with some other providers that I don't know. Now, for RNA specifically, I don't think an extraction-free method exists so far. So what has been developed, especially for blood, is all these methods around sampling small amounts and stabilizing even at 4 degrees or ideally at room temperature. But yes, there is no, at least not to my knowledge, a method that is already available to perform direct blood handling, or even with other liquid biopsy type, like what I mentioned, urine or saliva. Then I can give you one clue of when we developed Luthor, and when you have other protocols out there for especially single cell RNA sequencing. When you have very small amounts, you cannot extract RNA, because by the extraction will just, you lose everything during the extraction process. So the secret is basically the lysis. And all these protocols, and especially our Luthor protocol, the kit contains a lysis buffer. You can also download the manual, you will see there is a lysis buffer. Well, the composition we cannot disclose, but essentially it's to lyse the cell. So the secret in the end is to adapt this kind of lysis buffer to be efficient with any kind of sample. It can be with FFP, it can be with cells, it can be with blood. This is the first question we need to answer, and we're working on it. And the second question is, even if you find a way of lysing these samples, and I'm sure you can find these in the literature, because basically you use detergents, chaotropic agents, all these kind of chemicals that are well described in the literature. The second question that may be even more difficult is, since all these kit solutions workflow, they use enzymes, and these enzymes have special requirements in terms of salts, pH, osmotic pressure, etc. You need to have a lysis solution that does not inhibit the subsequent reaction. And this is probably why such solutions don't exist yet, at least for large amounts of sample. For small amount, like this one cell, no problem, we have this solution. So that's the path we are trying to follow. And yes, if this is of interest to you, don't hesitate also to contact me, or and then when we are ready, when we have something that could be interesting, maybe we can catch up. The last thing I want to mention is that we talk about liquid. Sometimes we can also work convert, let's say, a liquid into a solid, and this typically with blood is the blood cards or the FTA. This is also an interesting way of preserving the blood without having to carrying around liquids, especially blood. You know, the tube can break and you have stability issues. And then the challenge remains the same. When you receive the card, you need to extract what is in the card. So we're also working on these solutions to work with blood cards. And blood cards are super easy because essentially, in terms of collection, because you can be at home, you take a finger prick, put on a card, ship it by post, and that's it. So this is also the future probably.
Thank you.
Thank you very much.
Thank you. Lubanga and Filippo, the question, very interesting answer, well tackled. Thank you. Any other one from the... I am also checking the chats. I haven't seen any question. So please, there was a question from the participants reserved before. A participant is asking whether you have a method to analyze glycoRNA at the moment since it's still emerging. Then another participant asked what are the collaborative platforms available in RNA research to foster hands-on training and skill development of African early career scientists in life sciences. He's also asking if it is possible for researchers in Africa with academic background in other biomedical sciences, sciences like anatomy and physiology to be trained and guided on different areas of RNA research. I think from the questions collected, these two need to be discussed. Thank you.
So for glycoRNA, we are not working on any solution for now, but I'm just writing it down because this is also important as a feedback from users, from scientists. If there is an interest in this field, we collect this interest. And if we have repeated requests, then we will surely focus on it and develop something. So unfortunately, I cannot say we have something, but maybe in academia or some other participants here have ideas, but not at Lexolent, but I write it down.
Okay. I would want to share an insight briefly because in our just concluded RNA, we hosted a guest who is actually working on glycoRNA, and our team, we've looked at some considerations. You find out that glycoRNA is about glycoimmunomodulation. So in glycobiology, you look at the detection of expression of that RNA to answer a question on whether the glycosylation of mRNA mimics the memory of glycation induced by HbA1c. So we are trying to look at that concept because now there is a discussion about type 5 diabetes, which could be linked to a kind of epigenetics because it's about a post-translational modification. So if we are able to discuss glycation in line with glycosylation, because glycation is just a priming and maybe it cannot be inherited because it doesn't involve enzymatic process. But glycosylation, due to the presence of Siglec, that is membrane immunomodulators that link to the glycan moiety attached to a species of non-coding RNAs. So at that point, there may be chances of developing a kit to detect the glycosylation level after a moment of diabetes so that we can talk more of how these diabetes are inherited along epigenetic line which HbA1c currently used to diagnose extent of diabetes and diabetopathies cannot explain, because it doesn't involve enzyme activities, but glycosylation involved. So if there could be a way to analyze glycoRNA, maybe pre-treatment or post-treatment, that will also be discovery of new therapy. So I am recommending that maybe your team can look at that, and we've currently submitted a opinion paper to RNA biology. So hopefully, if they publish, it could still form part of our collaboration with your team. Thank you.
Yes, thank you very much. And in general, we are also looking at trends, and typically Professor Ross mentioned the RNA Society. We have scientists going there as well to see what's going on, what is the new trend. And RNA modification, it's very wide, because actually DNA modifications are not that many and relatively easy now to analyze, but RNA can be modified in many more different ways. And unfortunately, for many of these ways, we need different solutions. So
exactly.
yeah, let's see. But I will mention this to our scientists, who are certainly more aware of me than me, sorry. And yes, we can also keep in touch later on for this.
It's okay. Do you have any comment on the second question? The collaborative platform available for RNA researchers in Africa to foster hands-on training, and whether scientists from other biomedical sciences like anatomy and physiology can also be trained for this. Do you have comment on that question too?
No, I'm not aware of. Well, of course, in the internet you find like RNA-seq blog, you have encyclopedia, so you have a few websites that basically summarize news technologies that are also sometimes sponsored by companies like ours. So yeah, the web is usually the best, but then for hands-on, yeah, I'm not aware of such a platform. Also with video tutorial, I have not seen them until now. Unfortunately, yeah, this means probably, and maybe that's the best solution, is really to encourage collaborations with, of course, other labs, where you can also obtain grants to travel and to train on site with dedicated people, and to stay for a certain time. I would really recommend this. Sometimes we also see, when you prepare NGS libraries, is maybe the first shot you're lucky, but then the second doesn't work. So it's very important to really, for really people who have no experience, for people who are molecular biologists, of course, we can also help you remotely. We can give you some tips and tricks, and you will immediately understand, because it's also about working with RNA. Sometimes we see labs, they have no practice with RNA. And as we know, RNA is very unstable, and you have RNases everywhere. So you need really a clean lab. That's really very important. And sometimes it seems obvious to say, okay, yeah, my lab is clean, but actually not for RNA. And then the second thing is really to gain experience. So probably by collaborating with some labs on some project, or maybe some companies, and spend some time and really repeat the experiment several times before you really know that you can reproducibly prepare libraries. And this can also be a source of variation. Maybe you have learned, you have seen it once, and then you go back to your lab and it doesn't work. Why doesn't it work? So in this case, you can write us if it's one of our kits, and we can really try to help, because we troubleshoot almost every day. So we are really very experienced at tech support. But it's, again, yes, I would really recommend someone to if possible, to really foster collaborations with other labs who are experienced, and then get trained there. Yes. So unfortunately, I don't have a complete answer. Yes.
You have shared an excellent insight. That is exactly what we want. We have been on it for over 3 years now, and I believe some of our scientific community are gaining insight, and we are doing a lot. We're looking for collaborations, then opportunity for fellowships, because a lot of young African scholars have developed interest in RNA research, and we are just happy about it. But resource availability and funding has become limitations, but we believe we navigate it with time. We are very grateful to have you. I hope, are you leaving or you will still be with us? Because we have few other things to discuss then, like we are still in general discussion. Maybe you can still be with us for a moment.
Yes, it's no problem.
Okay, thank you. Please, the audience participants, if you have any question, again, you can raise it. If you don't have a direct question, I will call on Professor Patrick Madabuchi Aja. He's currently Dean of Research in Kampala International University, and he is the Director of Mobilization, of Resource Mobilization in African Life Science RNA Salon, for him to get ready to address concerns. If you have any question or things you want to discuss with African Life Science RNA Salon, just a concern or maybe objection, suggestion, then you can speak it up, let us address it. Then he will also give us tips on our future plans, and from there we will have recognize some persons that is in our midst from different parts of the country or the world, and we proceed from there. Thank you, Professor Patrick. You can unmute yourself and proceed.
Thank you, Dr. Peter. Please, if you should pardon me, I'm having another program which coincided, and in that program, I'm one of the guest speakers, though I've made my presentation, so I had to excuse them and went to somewhere to stay so that I can participate in this one. And I will also go back to conclude the other one. Well, in a special way, I thank Philippe for his wonderful presentation. You can see the extent RNA research has gone, and it's something that Africans should embrace. And one of the questions I got from people that have sent out their question before now is how can RNA research, or how can some of the RNA research outcome like the therapeutics or diagnostic can be implemented in Africa? And one thing is that the COVID-19 vaccine is an RNA research product. And many other vaccines that has to do with cell therapy has RNA technology embedded, even diagnostics. In recently, I supervised a student that developed an aptamer, that used an aptamer to develop a diagnostic tools for detection of Mycoplasma bovis. And we also have done a validation on that using using the...
Please, Professor Patrick, sorry for interruption. I just want to tell us that Professor Hyde has just joined. So when you finish, we will take his presentation.
Well, let me quickly round off so that he'll come in. Then I can come after his presentation to finish up my responsibility. So participants, we have a lot of concern to address. As a member of Africa Life Science RNA Salon, we have our social handles, both WhatsApp, both Facebook, X handles, and what have you. So you can follow us up there. We have a lot of information, a lot of connections, even on postdoc and benchwork opportunities that scattered in Europe. So let me give the opportunity for the second presenter to make his speech. Thereafter, I will come back to finish up my own assignment. Thank you.
We are happy to have you, Professor Hyde. Professor Hyde is an associate professor of pathology in Harvard Medical School and director of Precision RNA Medicine Core, Bethel, Israel Deaconess Medical Center. He will be giving us a guest speech on translating RNA biology, a system approach to precision medicine. Professor Hyde, thank you for making out your time to be with us. We are happy you are here finally. In short, we waited a lot, and thank you for being timely. You came, but at the right time. You are welcome, Professor Hyde. We could not hear you. We can't hear you now.
Can you hear me now?
Yes, excellent. We can.
Yes, we can hear you.
Okay.
Welcome.
Please accept my deepest thanks for this opportunity to join my fellow Africans. As a proud South African, I'm glad to be here. Now, today, I want to say, first of all, thank you. I think innovation is a powerful thing, and you are innovators. Thank you for letting me join you today.
You're
Should I
welcome,
start my presentation?
Yes, you can.
Okay. Okay. I'm going to do a check. Can everybody see
Yes, we can.
this window? Full window, okay? So now I'll start the presentation, which will be about translating RNA biology. Look, I've done some work with RNA in the lab in 1986, 87, 88, 90, 91, 92. And I became fascinated with this concept that there was a thing called a transcriptome. Many years later, I realized that there are parts of that transcriptome that yield secrets of biology. And that concept that you can find biological secrets with just RNA, that's hooked me. And I've been trying to make discoveries with that since that time. So today I want to talk to you about a real problem and no solution, but an attempt at a solution, which is what we're trying to do. The real problem, now is this working? The real problem is that we don't have ways to target drugs effectively. The genome and our biology is very complex. So when you're looking at a complex disease like Alzheimer's disease, you can use genetics to find specific genes that tell you a little bit about what that disease might be doing, and you can target those genes. And frankly, that's a challenge, because which gene do you choose? If, like you and you're curious, and I had the opportunity to spend one day a week in a local biotechnology company. I jumped at the chance. That's really interesting for an academic. And I sat in a room called Target Day Room. And it was filled with all of the top scientists in that company, and they clicked on click machines as presentations were made throughout the day about each of the potential targets for the drug. The highest number of clicks was what $200 million was spent on. And let me tell you, that trial failed. So I learned a lot through that process. Currently, in Alzheimer's disease, we attempt, apparently, I mean, we meaning the field, not myself, but the attempt is to clean away the plaques that are thought to cause Alzheimer's. And even if you have antibody-based therapies like donanemab and lecanemab, which are safe, their efficacy still is not that much. And that is because we don't really understand when to give them to people, how to give them, and I don't think that's the whole picture. So we need more effective targets, as I started with. And that's a real challenge. How do we go after that? So if we're going to have new strategies, we must make a plan. And my plan has been to think about using RNA as a medicine. So to get there, we have to understand when and where it's active. And the initiative for RNA medicine, created by Frank Slack, who was the co-discoverer of the microRNA Let7, for which the Nobel Prize has just been awarded to another colleague, Rafkin, at medical school. This guy is another South African, Frank Slack, and he created this institute to make and translate RNA discoveries into novel therapeutics and diagnostics. And honestly, we're all trying to do that. And I want to give you a couple of examples of what we're doing today. So I'm going to work today with you on microRNAs. And because you're looking at RNA therapy, everybody knows what they are, but we understand that they were discovered in Cinerabditis and that they're tiny. And I love them simply because they're ubiquitous. They are known regulators of gene expression. They're ubiquitous, and they're a lot more precise than transcription factors. And that is the key. So if we look at the mechanism of RNA function, we can understand that these are created, as all RNAs, in the nucleus and processed by specific complexes to result in a mature microRNA, which eventually within the cytoplasm is incorporated into the RISC complex that guides to mRNA via base pair. Now, the key here is that with that complex, then you can do repression of translation or induction of mRNA degradation. Repression of translation. So this is a translational event, which means that the mRNAs can be produced ad nauseam within the cell without feedback, theoretically. And then that mRNA that exists in the cell is then translated. And during that translation event, you're getting modulation. So as these guys are ubiquitous, they mark something. And I'll go through that, but they mark processes. And I love that because I work on pathways. So pathways, processes, microRNAs, these things are adjunct to each other. It makes it easy for me to understand. They may represent good therapeutic targets. So we might be able to put in microRNAs through encapsulating them in some system and wrapping them in safe structure and getting them into the cell. In other words, wrapping them in safe structure means a nanoparticle or means modifying them so that they mimic the actual microRNA, but they're more inert as they get into the cell. Also, by their ubiquitousness, we can use them for diagnostic assay. They modulate systems, and that's why I like them. RNA muted, they represent a novel class, they're systems level. And I think that they are interesting because if you can perturb them, you can look at their impact on the system. So it's almost like using, if you pardon me by saying this, using the human as an experimental model. I have discovered or co-discovered 4 microRNAs, and in each case, something fascinated me. Every time we looked at the genes that were the targets of these microRNAs, I noticed that the genes and their products, the proteins, were interacting with each other far more frequently than I would have expected if I just looked at a random set of genes. And that fascinated me because that means that through evolution, these proteins, which have been interacting with each other, and their gene products, so these genes effectively that have been interacting with each other, have found a modulator, or that modulator has found them and has been selected. So that process of interaction is ancient. And that process of interaction is also functional and regulatable. You can see where I'm going with this. So what if we knew the microRNAs that coordinately regulate these disease pathways? So this is work I'm doing with Frank Slack and my colleague Du-Young Kim over at Mass General. And we're trying to find microRNAs that modulate Alzheimer's disease associated pathways. This is no easy journey. And I'm giving you a high-level overview, which is a lot more fun, and a short talk, which should be good, so we can keep your attention. So this is the bioinformatics slide. So those of you that hate bioinformatics, you can close your eyes, but I won't take long. And what I want to show you here is that we have, oh, excuse me, we have, I'm just trying to get a marker, I can't do it. What I want to show you here is that we have generated a method where we look at pathways by their activity. And in panel A of this slide, you can see that what we do is that we simply take a gene expression profile. And that can be count-based, that can be in old array, it doesn't matter. But the point is we take this gene expression profile for a single sample and we rank the genes in that sample. And that allows us to say we have a top-ranked gene and a bottom-ranked gene. Then what we do is we put these genes into a mask. And we take, for instance, the KEGG pathway corpus, a few hundred pathways. And we ask the question, is any member of this ranked gene list that I've taken from this sample for its expression a member of a pathway? And if so, what is the rank order of the gene in that sample? Can I take that rank order and the gene name and put it into that pathway mask? So for each pathway, we get a set of ranks. Now you get where I'm going, because if you have a pathway with a lot of high-ranked genes, it's likely to be more active than a pathway with a lot of low-ranked genes. And you can see an example of that in the slide C, where you see, well, part C here, where you see cases versus control. Here's a specific pathway which is less active in the control than it is in the case. What we can do then is we can ask, what's the activity of that pathway? You see case and control here, but imagine if we took the controls and we passed that pathway across all of the known experiments that we could find in GTEx, which is a background that is for count-based sequencing. And over those hundreds and hundreds, and well, thousands actually, of experiments, what we get back is the relative co-expression of any single pathway with respect to another pathway. So in sample D here, you see a pathway co-expression network. And what we do then is we cluster the network in E, and we find pathway co-expression modules. I hope you followed me through that, because all I'm doing really is saying, are there groups of pathways? Do pathways group in any way? Well, this is a canonical situation. Pathways group here as they would for ancient history, for evolution, and we can map them together and say, of the pathways that we see activated, do any group? Yes, they do. And if they do group, do they group into clusters? Yes, they do. And if that is that cluster then regulated by a set of microRNAs? And in panel F, we look for those microRNAs. What we actually do is we look for the targets, known experimentally validated targets of those microRNAs, and we ask, are they enriched in those pathways? And obviously we compensate for pathway size and such. And we've written up this method, and there's tools available in Bioconductor in R. Hello? Okay, I'll continue. Tools available in Bioconductor in R through the Panomere system and package, and you can see publication at the bottom of this slide. Okay, next slide. Let's see if I get this. Okay. So if we look at these clusters of pathways, we find that the largest cluster has enrichment for processes in amino acid metabolism, but interestingly, do you see a lot of nucleic acid metabolism and processes? We often see this downregulation of nucleic acid-based metabolism in AD. A lot of trouble at the DNA level, basically. We also see in the second largest cluster, which is upregulated, a set of pathways involved basically in response to inflammation or inflammation, and also pathways involved in plastic, what I would call remodeling, angiogenesis potentially, all aspects of response to some form of infection. But really what's intriguing here, this is AD, that we may be seeing signals for cellular remodeling, but that's a very controversial area. In any case, we've got clusters. What we do then is we look at the top predicted microRNAs in those clusters, and we find that there are similarities, first of all, in the microRNAs between two different regions of the brain. And also what we find is that there's an enrichment in those microRNAs for known microRNAs that are impacting patients who have Alzheimer's disease. So that's great. We're able to make predictions. Are those predictions real? We're not really aware of whether they are real or not, but what we are aware of is that there are correlations between those predictions. In other words, if we take a Jacquard index, which is basically a way of asking, is there overlap, and is the overlap relative to the total size of the overlap? What is it? And you can see basically that there's quite a high degree of overlap between the regions of the brain. There's also in this annotation something saying AB42 cluster. That represents a cluster of expressed genes and pathways that come from a model of Alzheimer's disease that we have generated. And what this slide shows is that both models and regions of the brain show high concordance, as much as 50% in some cases if you look at the TCX versus the A-beta-42 cluster. So this means that we know something about microRNAs in that they appear to be predicted as being common in a large number of different complexes, systems. Finally, looking at this bioinformatic type of proof, if we look at the differential expressions of a known microRNA that we've discovered, and we look at the genes that are targeted by that microRNA and ask the question, are they changed in expression? They are. And in actual fact, they're changed significantly. So that's about all we can do at the predictive level. But it's powerful. It gives us a means to prioritize unbiasedly a targeting system. So I'd like to close that section and then explain to you a couple of experiments and then finish off. If you want to look at what I call human readouts, that's cool because it means we're touching humans and there's something real. It's not animals, it's real. But as you know, working with human data, humans are very, very variable in the way they behave. So we have serum that we have requested from a public resource called the Religious Orders Study Memory and Aging Project. And actually, these are the tissues and brains and blood of nuns and priests. And we very much, of course, respect that, but it's fantastic data. So what am I saying here? I'm telling you here that over the last 2 years, we've spent an enormous amount of time being very careful to figure out what microRNAs are in the blood of these subjects, some of whom have Alzheimer's disease, some of whom do not have Alzheimer's disease, some of whom are over 100 years old, and some of whom, independent of their age, are riddled with pathology from Alzheimer's disease, but have no cognitive loss. And we call those subjects resilient. We'd really like to know a lot more about those subjects. So we've looked at the serum of these guys. We've also extracted exosomes from the blood circulating from the serum. And let me assure you that that is no trivial process. It requires taking a column-based approach. It requires a lot of expertise. And then we sequence the microRNAs in these exosomes, once again, using a PCR-based approach. And we've taken the whole serum of these microRNAs and performed small-seq upon them. And we've also looked at the concordant brains, the same brains of the same individuals in the dorsolateral prefrontal cortex, using data which was generated in 2013 by the De Jager group using an array. And I haven't listed it here now, but we've also now looking at some count-based data from the same subjects for sequencing of those brains. So we have a large number of different kinds of technology converging on signal. And this question I have for you is, do you think we got a signal? And the answer is yes. when we've compared those assays together with exome assay, we found one microRNA that was concordant and dysregulated across all of these body compartments. I'm really excited to say we're also looking at cerebral CSF of the same subjects, and we're looking there to see if we can find that same microRNA. If we can, what intrigues me is that microRNA is unique to subjects who are resilient to Alzheimer's disease. If we can find that microRNA in the circulating blood and reproduce that across thousands of subjects, so it only appears in those subjects who are resilient to Alzheimer's disease and have no cognitive loss but have pathology, then we have a marker. And it's a good news marker for those individuals who have what I call the bad allele, ApoE4. ApoE is a large gene, which, as you know, is deeply involved with Alzheimer's. And the ApoE4 allele, if you're homozygous for that, at the age of 57 and onwards, you'll start with mild cognitive impairment, and by 80, you'll be dead. So it would be good to know, for instance, if you were a subject which was resilient with those particular genetic background. So you can hear from me what our strategy is. We've used a tool called Panamia, and on the bottom right here is a means, if you photograph that, you can go ahead and find the Panamia publication and download the tool. And what we think is that that tool yields high fidelity microRNAs with hypertherapeutic potential. It also lets us know if we look for biomarkers, you can reverse engineer that signal and know what pathways are being impacted and know what processes are being read out. I run, or oversee actually, a lot of other people run the Precision RNA Medicine Core at Beth Israel Hospital, where I work. And actually, that's made up of a number of cores. One is a spatial technologies unit. Frankly, in that unit, we're still learning about how to trap microRNAs in a spatial way. We don't do that yet. We have a non-coding bioinformatics unit. Interestingly, we have an RNA delivery unit, so we can package RNAs into nanoparticles and deliver them. And we have an RNA detection unit, which has allowed me to do the presentation you've seen today. The website is down, I've just checked, but noncodingrna.org is the overall website for this system. I'd finally like to finish with another sort of evidence presentation here. And my colleague, Jan Heng, who manages the RNA detection core and also is a faculty member at Beth Israel and Harvard Medical School, she's been working on plasma microRNAs. And she's discovered that if individuals are treated with cisplatin, this microRNA is linked to a good response to cisplatin. And she's in the process of performing a correlative analysis with paired tumor microRNA. Once that's complete, it's possible, as you can imagine, to move forward with developing a diagnostic. So I'd like to thank the amazing people I work with. These are just the PIs, but it's just a stunning group. I really enjoy my work, and I encourage you to look in deeply into this area. It's fun. Anybody interested in my work, take a look at the website. You can also find that by going to this QR, and I invite questions. Thank you very much.
Thank you so much, Professor Hyde. We are grateful to have you today. So I will call on participants to ask their question. Please, if you have your question, you can unmute yourself and ask, or you raise up your hand so that I call you up. Dr. Tusubira, you can proceed to ask your question.
Thank you very much. Just a minute, I switch on the video. Yeah, I'm Deus Tusubira from Uganda by University. I know Professor Hyde was introducing himself as an African from South Africa. Glad to meet another African. And that's so nice, yeah. You know, when we started this Salon series, we are looking at Africa as a whole. The biggest challenge we have in general, most of the sub-Saharan Africa, people tend not to have so much ambition in this area of research because of we feel the facilities are limited, especially the facilities. But I commend everyone and the present especially who have now inspired. Now we hope we shall have a big team of those people who are interested in especially RNA research. When you are exploring this problem, especially for with Alzheimer's disease, aging is also becoming a big challenge here in Africa, if you didn't know, okay, in general. So these problems are becoming more prevalent, and we feel this is area of research is becoming more and more important in our communities. And so we need this kind of work to be done more. And for me, I don't have a specific question, but I'm interested in the following. You are pursuing a big challenge. You mentioned how the beauty of microRNA fine-tuning, you see, that crosstalk with the, would I say the phenotype, the protein, and then around there. And then maybe we also know that there's a lot of crosstalk. I would say crosstalk somewhere. It looks a bit complicated. But I love the beauty of the way you approached it until you're looking at system-wide. So you see, there's a lot of crosstalk here and there and there and there, but you are looking at system-wide. So the omics era, large-scale things and such things. So now, this is just thinking about what you have done and the beauty of it all. What I want from you, which would be a question, is to give some advice on the young people. In these RNA salons, we are mostly having many young people who are in their early careers.
Yeah.
They are looking at these big ideas.
Yeah. And they're asking, how do I get there? How do I get there? Right? How do I get there?
Yeah. How do I get there? They don't know where to start from.
Yeah.
They feel like, oh,
I know that
up.
question. Yeah. Thank you, Dr.
Some advice to the young people.
You have given a good reflection, of what I described, and also in a gentle way, you've described the motivation and somewhat frustration, right? You want to get there. How do I get there? So for me, it's like looking at a car. How do I buy that car? There's no way I can afford it. I don't even have a road, right? We've all got a road. And what I want you to know is that together with your colleagues in Uganda, everybody across Africa I've worked with, we created a bioinformatics network from nothing, no money. The WHO said, create a bioinformatics network. We'll give you money if you do it. And after many years, we got $50,000. That's it for, 3-year program, right? But I want you to understand, it wasn't the money. It was just that we were prepared to talk to each other and grow together. And we brought in some experts, but we had some experts locally, blah, 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 blah. We built a network. What that network did was provide training. What you're doing today in the RNA Salon is provide training. And I'm really impressed because everybody from everywhere is on this call interested in RNA, right? So what I found was the most powerful engine was workshops. Now, I don't know if there are any RNA training workshops across Africa, but if there aren't, that's okay. Let's start thinking about creating them. It's not impossible, it's easy. What's difficult is getting organized, right? But what I want you to know is training workshops is what I regard as the first step. The second step is to go to a center where there is somebody interested in your problem in Africa, who has colleagues around the world who are also interested in RNA. And that could be any of the fine scientists on this call. There are many doctors and professors, and we care about RNA, right? So yes, you go to that person and you find a workshop, and that's a catalysis, and you begin. So you train with that person and you learn through the workshop. What the workshop does is you meet other people in the same boat as you, and now you have a cadre of good people to work with, and you've met somebody who knows about the field. That's the beginning. You don't ask for favors. What you do is figure out a way to train with those challenges, even if you're training in bacterial biology at a plant science institute, you're still interested in RNA and you keep that interest going. You find a way to attend workshops. That's the key. And you get attendance certifications, etc. With that on your CV, you can then start applying for internships. I offer an internship, which unfortunately, as many of you know, I'm in America now and there are some challenges to bring people, okay? But we're already thinking, how can we make a virtual internship? That's the next step. You gain association with people who have funding. So that's usually overseas, and that's usually, but not always, outside of Africa. But I'll tell you, there's some good funding in Africa too for training. Okay, and all of us look for that in Africa, as you know. And then if you can, and there's a good center in Africa to study, you go there, or you hybridize with another site overseas. And don't ignore Asia, Japan, China, all these places have great study opportunities. And then most importantly, you find money and you come home and you influence science at home. I'm telling you my story. But do you understand? It's on you to find those workshops. Okay. I'm sorry I don't have a more powerful answer, but that's how I think we can do.
I appreciate.
That sound well appreciated, Professor. Thank you.
Yeah. Maybe something small. I like the way you have these groups categorized. I'm thinking of that connection between the gene, then in between, then the phenotype. You see the proteins, the disease.
Yes, we call that the intermediate phenotype. Between the phenotype and the gene is the intermediate phenotype. It sounds great. It means we don't know what it is.
Yeah, so that is it. Because it's kind of fascinating to me. Many people, they have these genes, they have these so many things happening, but they're okay. You see? So it becomes really very complex to study. It looks really kind of intriguing. You see, you talk about resilience individuals. That's where I'm coming from. Maybe you can talk more about those individuals.
Well, I'll finish off, and I think, Dr. Tsubiri, we need to give other people a chance to talk, but I'm really keen to talk about those resilient individuals, and I will say in 1 minute, because I believe that they are our future and that our study should not be about disease, it should be about health. How do we stay healthy? And that's not about wellness and pills and drugs and, whatever, dacha. This is about finding the mechanisms inside us that protect us. And already we have found in centenarians, which we have brains of in our freezer. We have 12 of them in our freezer. And we're looking at these centenarians, as are other people around the world. And we've discovered that some centenarians who are resistant, they don't even have pathology, have a system in their brains which works more than our systems and normal people. And that system is an engine to clear A beta plaques. Just as you hear that the antibodies do for the drug therapy, their proteolysis systems work more. That's it. That's all we know. Next question.
Thank you, Professor. Please, other questions, please, the participants, please, you can ask your question. We've longed for this opportunity and we have it today. Prof is here with us. And even if you have more for Dr. Filippo, here we have brought academia and industry together to discuss perspective in RNA. So it is a good setting to talk. And Professor Hyde and Filippo, I am happy to tell you that African Life Science RNA Salon is 3 years today from inception. And over the years, we have organized around 6 in hybrid meeting and 2 transnational webinar, today making it the 3rd transnational webinar. And in RNA Society, we have had a recognition even in RNA therapeutics being organized by University of Massachusetts, Omar Chang's lab. They also recognize our impact. We also had Sister Saloon in Kenya, RNA Kenya. So we have been working together, and the joy is that Africans, young scholars are becoming interested in RNA research. And having you here is a happy. Professor Hyde, I'm happy to tell you that when Professor Prashant recommend you, he say, I am recommending Professor Hyde. He is fond of Africa. He's fond of Africa, and it will be good to talk to you. So we are happy. At last,
Wow.
you also made us.
I'm part of Africa just as you are, not just fond of it. Thank you for the
Yeah.
opportunity. And now I'm going to turn off my slides and leave it for the next presenter. Thank you so much.
Yeah.
I've really enjoyed it, and good luck. Peter, please contact me. We must discuss the RNA Society and connecting it to the operation we're running here at Harvard, okay?
It's okay. Thank
So
you, Professor.
let's chat offline. Bye-bye.
Bye. So, Professor Patrick, you can proceed. Please, Dr. Filippo, being around is also good to us. So you can be with us if you're still free till we are done. But we will soon be done anyway. Thank you, Professor. Thank you. Professor Patrick, the floor is open for you.
Thank you, Dr. Peter. Thank you, Professor Hyde, for your very insightful presentation. And this is on a good note, an African making wave in RNA research. That's a good one. So thank you. We have learned a lot from your presentation today. Well, I have the mandate to look at some of the complaints of the participants. And incidentally, some of the participants, some of the people that fill this form may not be present here, while some may be present. And some of the issue has been addressed, like the issue of glycoRNA methods. Then the method is not far-fetched. It's based on metabolic labeling of cells with clickable sugar precursors, such as N-acetyl-mannose-amide tetracyclates that can be added to cell culture and incorporated into glycan as a salicylic acid mimetics. These methods are there. It's a standard method. And there are also some concerns. How can this kind of studies be done in Africa? Actually, we have limitations based on infrastructures, but there are some upcoming laboratories, and with help of collaboration with European Northwest collaboration can also be very helpful to overcome some of the challenges with Africa. Africa, even if you go to Europe, you see most of the researchers there are also from Africa, like Professor Hyde, and even Professor Kahinde has been researching on microRNA. So there are a lot of hope for Africa, and most of the products we are using in diagnostics, even in vaccines, as a result of product of RNA research. So there is a need for us to open our mind and collaborate. Just like I said earlier, we have social media handles like X handle, we have Facebook, have even our website, also have a WhatsApp. Most of the times we advertise some postdoctoral opportunities, benchwork opportunities via these social media handles. So for some of you, the early careers that want to pick a path, a research path or interest in this area, can follow us on those social media. We're always there 247, and we'll have Dr. Peter as our social media handle experts who handle most of the things. Then we'll have Professor Deus and Professor Kainde. All of them are always active and will share all the links we have come across elsewhere that may benefit the members. So we encourage you, if you have not been part of our exercise, we encourage you to join us. So all these are social media. Apart from this webinar, by first week of September, we are going to have a hybrid symposium that's going to take place in Kampala International University, Uganda. And thereafter, by October, we are going to have another hybrid program that's quite captivating that will take place in Nigeria, in Boy State University, Evangel University. So, and we ask you and inform other people to be participating, because those programs are going to be hybrids. They're going to be hands-on. Now, the case of Kampala International University, we are going to partner with Institute of Biomedical Research, IBR, to see how we can have some hands-on on basic knowledge of RNA research and to consummate our hybrid symposium. So I ask you to stay tuned. So thank you, Peter. So if there is any questions from the participants, they can come up with them.
Professor Patrick, for the addressing the concerns. For participants who still have concerns or question or comments to ask, to add, they can. The floor is open. We are on general discussion now. And of course, you know, in RNA Salon, we value inclusivity. So we need women to comment as we proceed. Please, Professor Hyde, Professor Prashant Surajavala extend his regards to you. He was here before he left. But he's currently attending a meeting in Florida. That is why he could not continue with us. Please, let's hear more then. We want women to put their voice. Thank you. Okay, Dr. Esther, you can talk to us. At that point, then I think Apollonia, you can also comment after Dr. Esther has talked. Thank you.
Yeah.
I mean, Dr. Esther.
Thank you very much, Dr. Agu. I am very happy to be in the midst of reputable scholars, especially our very Professor Winston. I have really enjoyed the teachings from all the presenters, and I can't wait for the upcoming events. I'm highly happy to be part of this great research umbrella. I wish to thank the organizers, especially Professor Patrick, Dr. Ago, Dr. Tusibra, and all the founding members. Thank you very much for this great opportunity that you brought to Africa. Thank you very much.
Thank you, Dr. Esther. You did not tell us your affiliation, or you just, so you can.
Yeah, I'm sorry about that. My name is Dr. Esther Ugalo. I am a scholar here in Uganda, Kampala International University here in Uganda. I'm the head of research publications at the main campus of the university. Thank you.
Excellent. Thank you. We value women in science. You are representing well. Please, Ms. Apollonia Fujans, you can unmute yourself and also comment so that we proceed. Thank you.
My name is Apollonia Fulgency Yudas from Tanzania, currently a graduate student at Southwest University. I am honored to be here and listen to a light. And to be honest, today I have gained a lot, especially about RNA. Actually, I was performing same experiment today. So I got a lot, and some information and idea will help me to troubleshoot my experiment. I'm looking forward to learn more from you people. I don't have much. That's it. Thank you.
You're welcome, Ms. Apollonia. It's been a moment with RNA since I joined the lab with her, and she's doing excellently in qPCR. So that is why I wanted her to talk. So thank you for your comment. Please, any other participant? Otherwise, I call on Dr. Deus Dedit Subira to give a closing remark and a vote of thanks. Thank you.
Thank you very much, Dr. Peter Chinedu. I hope I'm pronouncing it properly.
Excellently, yeah.
Should you say Chinedu?
Yes, excellent.
Okay. So we have come from far. Every single day we are becoming better and better. Most important is the engine to progressing this kind of group, research group. And Dr. Peter has been like the glue, keeping everything moving. Most of us are old, we find ourselves tight here and there. Dr. Chinedu, I think you started, by the time you joined at first, you were doing masters, isn't it? Now
Yeah,
it's
we
already
started
doctor.
at the beginning of masters, yeah.
So the masters. Now, so we are expecting you to the next level. We are going to hear you, professor. You see? The same I'm expecting with all members who have been persistent enough to be part of this team. There are some people who have always been present from the first event, the second and second. So thank you very much. Success in life is about resilience and persistence. I know even in chemistry, Dr. Professor Hai talked about a group of individuals who are resilient. That is RNA, biologically. But we can be resilient socially. And that's how we end up getting what we want. We are looking forward to expanding this activity, this society. We are creating a platform across Africa. It's not just Uganda, Nigeria. I'm glad, what's the name of someone from Tanzania?
Apollonia.
This is Apollonia, yes. This is the first time that we are having someone from Tanzania. Please tell all your members there who are doing research in RNA, molecular biology, and this kind of work to join our team. Please talk to Peter and make sure that members come and join. Every time we meet, I remind you the following, that we are Africa-wide. This is the Africa Life Science RNA Salon. Life sciences, all of them. So we are looking forward to becoming bigger and bigger. We appreciate how busy everybody is, but we feel that the only way you are going to succeed, you see, you notice that now every country is looking inwards. Look at what's happening to USA, what's happening to Europe. All countries are looking inwards. But for Africa, what are we looking at? How are we going to be able to mobilize the politicians to invest in this kind of research? How are we going to create a platform for at whatever level? So this is a platform. The politician will recognize us, and in their decision making, probably we shall attract resources at whatever level, either on national level or on organizational level. In your university should recognize who you are nationally, and then maybe Africa-wide. I'm thanking the presenters for today. Peter, thank you for mobilizing Professor Hyde. And I feel we need to keep in touch. So I will keep working on. Filippo, you are still around. Thank you very much for your presentation. Thank you for explaining the practical part of it. Many times we who are in academia, it looks so theoretical. We just talk about things. For you, you are being practical. You say how to do things. You see? So you are so special. And you heard Filippo from Tanzania, how she appreciated the way you explained the real things, practical. You see? Thank you for your presentation. We are looking forward to you being with us in the next events. They have already told you we have 4 events every year. This is the second event. 2 more are coming. And maybe we pray that you'll continue sponsoring us so that we continue next year. Maybe we don't want this cycle to stop this year. We pray that next year RNA Society plus maybe Lexogen will keep supporting us so that we continue this cycle and keep the community bigger. So with all those few words, I don't want to declare this webinar closed. I hand over to the organizer, Peter. Thank you very much.
Thank you so much, Professor Tusubira. Working with all of you guys, all of you has been so much encouraging, from Professor Patrick to you to Professor Keinde. It's been wonderful, and we can only promise to do more. And we are telling our community members to always keep updated and keep being with us. Please, participant, we have just shared the feedback link. With the feedback link, we can get in touch with you. For those who opted for certificate of participation, through the link, we will get the exact name the way you want it. Please, thank you all for participating. Professor Winston, Dr. Filippo, Lexogen. Every time I got email from you, I read and I feel so happy because the content you are sharing is quite encouraging and nice. And currently, my work also involves I am a qPCR, where I am investigating the level of expression of micro RNAs in following after treatment and before treatment in the disease model I am investigating. So I've moved from general biology, biochemistry, now doing a second PhD in pharmaceutical science, and now I am investigating RNA expression proper in molecular pharmacology. So it's been wonderful, and I thank you all for the opportunity to work with you. RNA Salon, we keep sending, updating you our programs in all our social media handle. Thank you all for your participation. Thank you, Dr. Filippo. Thank you, Professor Hyde. Thank you, the team of RNA Salon. I will just quickly share my screen to show you, so you know the RNA Salon team in person before we proceed to close. So the RNA Salon team, which I have shared before, these are the team. We have Professor Patrick. If you go to our website at afrilestrnasalon.org, you will get it. These are our teams. We have Professor Kende from Liverpool John Moore University. We have Professor Patrick, Dr. Deus, Dr. Peter. So please always come to the go to the website as we keep sharing content, and we will improve. And also, we are looking for collaborations and opportunity to recommend our active community members, so that they will get opportunity to maybe travel and break the barrier of lack of resources, just as I broke it recently, and I am now doing my research related to RNA. So thank you all for your active participation. We look forward to having you present in our subsequent program. Thank you all for joining, and have a nice weekend. Bye.
Thank you very much. Viva Salon.
Viva!
Thank you.
Bye. Bye-bye.
Bye-bye.